Good morning, good afternoon, and good evening, everyone, and welcome to another edition of JAXLive. I'm your host, Rick Huntress, Director of Business Development and Commercial Strategy here at the Jackson Laboratory. Today, I'm pleased to introduce my colleague, Dr. Basile Siewe, Director of Business Development for West Coast Operations and Pharmacology. Basile's a trained immunologist and works closely with our pharmacology clients on efficacy and safety studies, particularly in immuno-oncology. We're going to have an exciting discussion today, but before we start, as a reminder to our audience, you're able to submit questions using the comment box. Please add a comment or send us a shout out to let us know where you're viewing us from. We always like to see where folks are coming from. We'll answer as many of the questions as we can at the end of our live discussion. It's very early in the morning for you. Good morning, Basile.
Good morning, Rick, and hello everybody in the audience. Thanks for joining us today.
Your microphone's a little muted, Basile. I don't know if you can move in just a little bit more there.
Sure. Does that sound better?
That's much better. Thank you. So we're going to talk about immunotherapeutics today, and there's been a lot of success in the clinic. Some of these drugs have had dramatic impacts on cancer survival, and the word cure is even being used sometimes. So with the approval of new drugs, things like checkpoint inhibitors and CAR T therapies, there's been a lot of activity in the biomedical community to build on these successes and then optimize or improve both the efficacy and the safety of these immunotherapeutics. So in the preclinical space, how have Jax and you been working with your clients to leverage the platforms that we might have to look at efficacy of some of these compounds?
Thank you, Rick. That's a very good assessment and a good question. Essentially, as you mentioned, JAX offers some platforms which have been critical in driving those immunotherapeutic agents to the clinic. So, as JAX, we have the premier gold standard immunocompromised mice, which permits engraftment of tumors and also can be humanized. So traditionally, those companies that actually leverage the platform where we humanize the mice with CD34 hematopoietic stem cells and a tumor. So this permits them to actually look at efficacy and also delve into really mechanistic studies. So this is what we would do traditionally. Now, as you pointed out, there's been a spur in moving towards really optimizing efficacy of these therapeutics and doing more also to address safety issues. So to meet that particular demand, JAX has recently been offering as well a different platform, which actually entails humanization with PBMCs.
Okay.
So with this platform, we have a platform which is really well suited for fast, affordable, yet relevant data, generates relevant data to assess the efficacy of immunotherapeutics in the immuno-oncology space. So you could also humanize this, as you mentioned, humanize these mice with the PBMCs and also engraft them with a tumor, very similar to what you would do with the 34 hematopoietic stem cell humanized mice.
Thank you, Basile. So we started out talking about efficacy, but it's been demonstrated in the clinic. These immunotherapeutics and biologics can also be associated with toxicity. So how are these humanized models being used to evaluate or determine the unwanted effects that some of these biologics can have?
That's a very good question again, Rick. So, and you're right. These immunotherapeutic agents, checkpoint inhibitors or T-cell agonists, they have always been plagued by toxicities. So the umbrella term they use in the clinic is cytokine release syndrome, or short for CRS, which is really characterized by high levels of inflammatory cytokine secretion and ultimately multi-organ failure. One could think about cases in the clinic with anti-CD3 antibody, the OKC3 antibody, or CD28 agonist with TG-Nero. They had a huge toxic effect in clinic with their compound. And also recently with some companies that have tested the efficacy of bispecific antibodies. So how JAX really moved their foray into this safety space is somewhat serendipitous. We had a study with an immunotherapeutic agent, and upon injection of that agent into humanized mice, these mice rapidly showed symptoms of CRS, and ultimately rapid morbidity. So it led us to actually wonder, as as good scientists, what might be the reason for this rapid decline in the health of the animals and ultimately morbidity, and which led us to assess and determine that it's most likely CRS. And we moved on to really invest a lot of resources to actually determine if one could leverage our PBMC humanized mice platform to also not only assess efficacy, but also assess if those immunotherapeutic agents would develop toxicities, and particularly CRS.
I love the idea that serendipity is part of discovery, because that theme always seems to play out. But in an environment where we already have lots of in vitro-based systems to assess CRS, I think some of those include the use of PBMCs. Why would an in vivo system be necessary, or what advantages might it have?
So there are multiple parts to that question. I think the first indication is the fact that there is no FDA approved preclinical platform to assess CRS of immunotherapeutics. And secondly, all those agents I mentioned earlier that moved onto the clinic and did cause CRS, they were assessed by in vitro platforms and even in vivo platforms in some cases. So traditionally, companies would use an in vitro platform and then almost often would move on to a non-human primate in vivo studies. So those immunotherapeutic agents, they all pass those preclinical platforms without any red flags, and yet really caused CRS in the clinic with some unfortunate effects. Talking about in vitro platforms, they have an inherent issue with variability. It's really different when you're culturing PBMCs on plastic as opposed to an in vivo system. And so that's really where the JAX PBMC humanized mice has really multiple advantages. For one, it's an in vivo system, so you're looking at the system as a whole. It's a more complete approach. And second, we could then effectively tie in the levels of cytokines which we observe upon injection of a therapeutic to the overall health of the animal. So in an in vitro system, you're getting cytokines, but one is not sure what those levels actually mean. Whereas in an in vivo system, we could quantitate cytokines and also assess the health of the animal. So we could look at the temperature. We could do a look at even a liver toxicity, really showing that we could tie in the CRS in terms of cytokine release to ultimately the health of the animal, which is what you would expect to do in the clinic.
I hadn't thought of that, Basile. I work with a lot of clients on efficacy studies, and we're trying to move them away from clinical assessments into biomarker-based readouts. But that's a great point, is that we can actually combine the cytokine data with actual clinical observations to help strengthen the data set that comes out of that. That's pretty neat. So when we run one of these safety studies, how might that differ from, say, the efficacy studies where we started, where we were looking at tumors and drug response, but now we're primarily interested in the cytokine response. So how are those safety studies a little different?
The safety studies, they have a very significant and critical difference. And this is the fact that we actually have what you would call a positive control. So we use agents which have been shown to actually induce CRS in the clinic. So really clinically relevant inducers. And if you want to assess if your compound would cause CRS, we would include those clinically relevant inducers. We use OKT3, TG1412, or bispecific combinations which have been shown to cause CRS. And we could then benchmark, your compound to actually those really controls which have been shown to induce CRS in the clinic. Whereas in an efficacy study, there's really no appropriate positive control since we have the issue with donor variability. But with the safety studies, we actually have inducers which have been shown to induce CRS in the clinic, and also we could replicate those findings in the humanized mice platform.
That makes perfect sense. So given the utility of this platform to look at the potential immunogenic characteristics of an IO agent, Are there other applications for this CRS platform beyond just looking at biologics?
That is really what I would call an exciting development in terms of making, it just goes to show how critical the humanized mouse platform could become. And this was really driven by COVID-19 or SARS-CoV-2.
Okay.
I think we're all aware, that's kind of an ongoing issue. And one thing that came up in the SARS-CoV-2 infection is that these patients that develop CRS-like symptoms tend to have a higher morbidity rate. So, talking to clients, they kind of push us to assess if we could also use the platform. So now instead of assessing if a compound causes CRS, we actually use our positive controls to induce CRS, and then now assess if a compound could actually modulate or mitigate CRS. And this really goes back to what I was talking about earlier, that CRS has always been observed in the clinic with immunotherapeutic agents, and the FDA approved one compound to actually address CRS in CAR-T therapy. So the anti-IL-6 receptor blockade antibody, tocilizumab, is FDA approved to actually address CRS in CAR-T therapies. So we kind of went along the same lines. Can you induce CRS with one of those compounds which have clinical relevance of OKT3, TGNR1412, and then come back now with a compound which would possibly have an anti-inflammatory effect, to determine if you could then use that to actually mitigate CRS.
This is a great story, Basile. It's the way you map it out. It starts with an efficacy study in an oncology setting, trying to shrink tumors. That transforms into a safety platform to look at the biologics themselves, and then it comes back around to an efficacy platform again to look at the ability of other compounds to modulate the CRS that's induced, and seeing all of that utility from one platform speaks volumes. As we're talking, we got a question from the audience. I'm going to read the question here. Is there variability between different PBMC donors in the assay? And if so, how do we control for that?
That's a really a great question. So essentially, for a good assay to mimic what you find in the clinic, you actually want to observe donor variability, which is what we find on the platform. So we have a cohort of PBMC donors which have been well characterized and pre-validated. So we have different buckets of responders and non-responders, again, which is what you would find in the clinic. So, in terms of running those safety studies, we have cohorts we could use, which have been shown reliably to be either responders or non-responders. So I don't think we want to control for it. We know that's something to expect. So in an experiment, we always include donors which have been pre-characterized to either respond and induce CRS or non-responders. So that's a really great question. Yeah.
It makes sense. And, over the course of my career, I think that's probably been the biggest change in the approach to preclinical studies in general is, we started from the perspective that there should be no variability between individual mice in a study or between different donors in an engraftment, to the point where it's actually now preferred because it's a better reflection of what's in the clinic. So that makes sense. Unfortunately, we're out of time. For those of you who had questions, please continue to submit them. But if you want to reach out, you can connect with the Jackson Laboratory through our Technical Information Science team, which is micetech@jax.org. We'd be happy to get you connected with Dr. Siewe and other experts in our immunology group. Thank you all for joining us. Thank you, Basile, for a great discussion today. And I look forward to joining you all at our next JAX live stream.
Thank you, Rick. Take care, everybody. Bye-bye.
Bye-bye.